Okay, so hello everyone. I think we are a bit late today. Thanks for joining again. It's been some time since our last session in January. So welcome back to our webinar series, Talking with Titans. So this is a webinar series organized by ANN, Aquaculture Nutritionist Network, and AIC, Aquaculture Innovation Center here in Singapore. So AIC, or Aquaculture Innovation Center, is a center of innovation based in Singapore and is being founded in 2019. So we just became 3 years old, and we are here to help the industry doing research, training, and also outreach like this kind of webinars. And ANN is a organization, non-profit, created in 2011. We have more than 6,800 members, and it's been created by Dr. Albert Taken and Kabir Chowdhury. They are the panelists for this webinar series. So this is a platform we can discuss different subjects in different topics in nutrition, in aquaculture nutrition. And today we are honored to have Dr. Margareth Overland from Norway. She has do a quick introduction. She has more than 30 years experience in animal nutrition. She has a bachelor's and master's degree from Montana State University and also PhD from University of Minnesota and the Norwegian University of Life Science. And currently she's the center director for another center of innovation in Norway called Foods of Norway. And one of the projects she's leading is the production of sustainable protein and lipid sources using microbial ingredients and novel technologies. So the panelist today, as usual, is Dr. Albert Taken, who is here. Is a bit late for him now, so we thank him for coming all this extra mile. And also Kabir Chowdhury, he'll be joining us shortly. He's in a trip in Philippines, so he's got a bit stuck in traffic in Manila, so he's rushing back to the hotel to join us. So we really apologize for the delay to start this webinar series. So as usual, it's about 30 to 45 minutes presentation from our guest, followed by another 45 minutes or so of Q&A and discussion. So for the attendees, please, if you have any questions during the presentation, type in the chat box, and then we'll be asking during the Q&A session. So I think without further delay, we can ask Dr. Margaret to start her presentation.
Well, good morning or good afternoon, everyone, and thank you for this nice introduction. And also thank you so much for the opportunity to be here to share some thoughts about how we in Norway deal with the raw material crisis. So I was asked to talk a little bit about myself. And I grew up on a farm in Western Norway on a lake where we raised pigs and dairy cows. And this lake was located near our neighbors that established one of the first aquaculture farm in Norway. And it was an ideal place to have an aquaculture farm because they have access to a very good quality water from our lake. And in this lake, we have a lot of trout and Arctic char eels. So naturally, a big interest of mine is to fish. And I would say in the mid-50s, my parents migrated to Montana in the United States. And for many years, I grew up hearing all these exciting stories about the life in Montana. So when I graduated high school, I packed my backpack and I took off for Montana. At that time, I'd never been outside of the country. I'd never been on a plane before. And it was a new, exciting life that started. The first half a year, I was working on different farms. I worked in the wheat farm during the combining. I was the truck driver, but I also got to drive the combine. Later on, I worked on a big pig farm in the US. And then I went down to Wyoming, where I worked on a cattle ranch with the cowboys. So at that time, I was working for room and board. I got some pocket money. I also, as a wage, got a pair of cowboy boots and a plane ticket to go back to Montana because I wanted to continue my studies at Montana State University. At first, I studied animal science, later on nutrition. I took my master's, as was mentioned, in nutrition, and later on, I went to University of Minnesota, where I continued the studies on nutrition, and I finished the PhD at the Norwegian University of Life Sciences. And I continued working there as a researcher at that time in pig nutrition. Then I left for the industry. I worked with this big company in Norway called Norsk Hydro, working on the first alternative to in-feed antibiotics for pigs in the EU, which gave me an interesting background from both academia and the private sector. Then I returned to the university and I took the lead as a center director for a center called Aquaculture Protein Center. It was a center of excellence, working on developing novel protein ingredients. And they gave us a very important platform, knowledge platform, that positioned us to get the new center, and that is the Foods of Norway. And so basically, I have been specializing in nutrition in the US on a pig nutrition. And then when I came to Norway, I made this transition to aquaculture nutrition. And this gave me the insight in a comparative background, and therefore, helped me when we were developing these novel feed strategies for the fish. So now I've been working with the fish for about 20 years. And I would also like to mention that our university has a very long tradition in aquaculture. In the 60s, we pioneered we domesticated the Atlantic salmon. And a few years later, we pioneered the development of the modern salmon feed together with the industry. And today, the salmon is our most important farm animal, you might say. So there's a lot of history here. And that's a little bit about me.
Thanks for your introduction. And so Margaret's presentation today will be a very hot topic now, which is the solving the raw materials issue. And she'll give us a perspective from Norway. So Margaret, you can start your presentation. Thank you.
Yes, how does that look?
Yes, great. We can see your slides.
Okay, so basically, in my speech today, I wanted to start with a short introduction about what is Foods of Norway, what do we do in the center. Then I will touch on the challenges in the aquaculture sector in Norway. And then I will go on and talk about how we develop microbial feed ingredients, how we bioprocess the natural gas as well as the biomass to produce these ingredients. I will continue to give some examples from our research on the nutritional value and how we also assess the health effects of these novel ingredients. A little bit about how we work with sustainable assessment, technical economical analysis. And then if I have time, I will also talk a little bit about insects, because we also do quite a bit of work with insects. And then finally, future perspective. Then Foods of Norway. It is a center for research-based innovation that joins research and industry. And the center here crosses blue and green sectors, and it combines agriculture, forestry, and aquaculture. And an important background from this center is all about food security and the need to develop local ingredients. And since the center started in 2015, this has become even more important today because of the big challenges we see with the climate changes, the pandemic, as well as political instability, and now the war in Ukraine. And we can no longer just rely on imports. So, our strategy is to produce more food with less land under more extreme climatic conditions, while minimizing the environmental impact. And in Norway, as you probably know, we have very little agricultural land, but we have access to large amount of bioresources. And therefore, we use biorefinery processing combined with enzyme technology to convert these to high quality nutrients that we then feed our fish to produce food for humans. An important biomass we work with in the center is the forest or the trees. It is the seaweed and also byproducts from chicken, pigs, and poultry, along with grass, which are all abundant in Norway. And then we evaluate how these ingredients affect the growth performance, physiology, health, and product quality of the fish. But we also work parallel with other farm animals. And the other thing we do in the center is to develop new methods that we can use in our breeding program for direct selection of fish with high feed efficiency, so they can better utilize our resources. So these are activities. And if you look at the center, it basically consists of a large multidisciplinary team of researchers at our campus, but they come from all over the world. We have with us several international partners, along with 19 industry and innovation partners. And this way, the academy or the university sectors work very close with the industry, and therefore we can take the results innovation further to commercialization. It's a good way to ensure technical transfer, you might say. And these partners, they come from both blue and green sectors. And these include the pioneer in seaweed cultivation, Seaweed Solution, the large forest biorefinery company, Borregaard. We have with us Lallemand, who is very strong in fermentation technology, along with feed and breeding company from blue and green sector. In addition, we have with us Denofa, who was Norway's largest importer of soybeans, that are now working with us on developing alternatives. So in this way, we have expertise along the whole value chain, all the way from the biomass to the final meat or fish products, you might say. And a little bit about the aquaculture and a little bit of background here. And as you know, this is the fastest growing food producing sector in the world, and it's playing an increasing role in supplying the world's protein supply. Today, Norway is the largest salmon producer, but in the global perspective, in terms of volume, the salmon production is quite low. But when you look at the revenue, Norway has the world's largest marine production, which puts us in the forefront when it comes to technology development in aquaculture, you can say. And at the moment, we are producing about 1.4 million tons of salmon each year and about 1.6 million tons of salmon feed. The salmon industry in Norway is projected to reach 5 million tons of salmon by year 2050. But to reach this goal, there are many challenges that needs to be solved, and a major challenge is then access to high quality feed resources. And here, because of the rapid growth in aquaculture sector, that puts large demand on the feed resources, including the wild fish stock, which are now under pressure. But due to intensive research at our university and elsewhere, the salmon feed has changed dramatically over the last 20 years, from being largely based on fish meal to today being quite a bit based on fish, plant ingredients. And in fact, if you look at the figure here, about 71% of the salmon feed actually comes from plant. But this can be questionable from ethical and a sustainable point of view, because this can lead to intensified crop production, which then puts pressure on the land and water resources, and it also leads to increased competition for human food. And today, majority of our salmon feed comes from other parts of the world. About 95% is actually imported, and that's problematic. That is problematic. But our government has said by year 2030, all salmon feed should come from sustainable resources, which means a lot for our research. We have a lot of work to do in a very short time. And if you look at the graph here, you see that the alternative ingredients, such as microbes, are present now in the salmon feed, but it only constitutes about 0.4%, relatively small amount. So in order to support further growth in the aquaculture sector, we need to develop sustainable ingredients with a low carbon footprint. And we need also to maximize the use of the existing ingredients that we have available to us. And the examples are insects and microbial ingredients such as microalgae, yeast, bacteria, and filamentous fungi, and marine sources on a lower trophic level, such as the tunicates and microalgae. And all these are examples of what we're working in Foods for Norway and spin-off projects. Then a little bit about these microbes, because there are now increasing interest in using microbes in aquafeeds, because they are highly nutritious and therefore they can meet the high protein demand of the fish, such as the salmon. And they are sustainable because they can convert low value biomass and waste gases into high quality nutrients without putting pressure on agriculture land. They also can be produced in large bioreactor, and therefore they can be produced year round, independent on climatic changes. And because they can be developed from these biomasses and waste gases, they don't compete with human food. So there's definitely a lot of potential in these microbes. And I wanted to share with you an exciting microbe that we've been working with for quite some time, which is bacterium meal that is produced from protein from natural gas. And this process, we're using natural gas to ferment and produce protein. This is based on the bacteria Metylococcus capsulatus. So gas here is used as the carbon and energy source. In addition, we add ammonia to the process, as well as oxygen and minerals. And this all happens in a loop fermenter. And then we harvest this biomass. We centrifuge it, ultracentrifuge it to remove excess water. And then the biomass is exposed to high heat for a short time to sterilize the product. And then we spray dry it, and then you end up with a brownish red powder with a very high protein content. And when you look at the nutritional value of bacterium meal, you see it contains 70% crude protein and 10% fat. In that way, it's very similar to fish meal, and it becomes an ideal replacement of fish meal because of these reasons. But bacterium meal also contains vitamin, minerals, as well as bioactive components that have shown to have positive effect on the health. So we've done a lot of work with bacterium meal. And here you see the amino acid composition compared to fish meal. It's of high value, but it's slightly lower in lysine. But at the same time, it is high in arginine and tryptophan. Continuing here, we have done a range of experiments with bacterium meal in different species, including Atlantic salmon and rainbow trout, with good results on both growth performance and health. And together with other partners, we also have developed cost-efficient methods to produce this bacterium meal from natural gas. And because of the lower natural gas prices, along with high fish meal prices and access to these methods that are cost efficiency, this technology has now shown to be profitable. So therefore, our innovation took a new, or reached a new stage where industrial actors took this further. And some years ago, Calusta bought this right to continue this technology. And they built a pilot plant in UK, and now they're planning on scaling up the production in China. And in 4 months, they expect that the first factory will be ready. So soon bacterium meal will be on the market. We wanted to also produce this in Norway, but at that time, I don't think the industry or the government was ready for this. It would be a different situation today for sure. So while natural gas offers new opportunities, another option is to use a biomass from the land and the sea to produce ingredients such as microbes. And when you look at Norway, one of our largest resource that we have available to us is the trees, spruce trees, for instance. But nearly half of our country is covered with trees, and this offers large opportunities for value creation, and such as using trees for feed. And the key to unlocking these resources is the technology to convert them into microbial ingredients such as yeast. And I'll show you how we do this. Because here we take the forest biomass, we cut it into wood chips. The tree fibers are then separated into lignin, cellulose, hemicellulose. The cellulose and hemicellulose is then converted into sugars by enzymes. And then we take the sugars, put it into a fermenter together with nutrients that we get from the animal, fish and poultry byproducts. Then we add special strains of yeast to this fermenter. We ferment this, harvest the yeast, dry it, and then use it as a protein-rich ingredients for fish and other farm animals. And working with the tree fibers is pretty tough. But because of a strong enzyme technology expertise at our university, we can easily now convert these tree fibers into sugars that we take then further into the fermentation. These enzymes are called LPMO, and what they do is that they break up the cellulose structures. So normal common enzymes can have excess, and then they convert this to sugars. And these sugars, through this process, is of very high quality. And this is then what we use to produce the yeast. And when you think about it, we are using only the forest byproducts in this process. And this is based on sustainable harvest. And all parts of the wood is then converted to a range of different products. And this is done together with the partner Borregaard. And first, they take out the lignin and they use them lignin for a wide range of high quality products, whereas the cellulose, hemicellulose is converted to sugars and then further to yeast that we feed to the salmon. And here you see we also are making use of the animal byproducts and get them into the value chain to produce feed and then human food. Further, when you have harvested yeast, you are left with some leftover green carbons. These are converted into biogas, and what's then left is rich in minerals, can be used as a fertilizer, and then you have a circular economy and thereby closing the loop. Okay, we have the forest, but we also have a forest in the sea, which is the kelp forest. And Norway has the world's second largest coastline next to Canada. Long convoluted coastline with access to cold and high quality water, which provides large opportunities to both cultivate and harvest seaweed. And seaweed is pretty unique compared to the land-based plants because it generates a lot of sustainable biomass. They do have a positive effect on the environment because they only need the sunlight and the seawater to grow. And they also bind nitrogen and phosphorus that are lost from the aquaculture farms. And they also bind large amount of carbon dioxide, and therefore they help prevent acidification of the forest. And seaweed can be used for a wide range of different products, including as a feed ingredient and as a functional feed source. But when you look at the nutritional value of seaweed, it's relatively low. So to use it and incorporate it into modern feed, we need to use modern technology. In addition to containing some protein and complex carbohydrates, these seaweeds also contain a wide range of bioactive components with documented bioactive function. The most common components are the fucoidan and the laminarin, and these can then be used in functional feed for the animals. So a little bit about how we work with this biomass. So we are processing the biomass according to a biorefinery process, where we make use of the entire biomass for a wide range of different products. So what we're doing at first is that we take out those bioactive components, such as the fucoidan and laminarin, and we use them in functional feed for fish that we're working on right now, as a matter of fact. And then the rest of the biomass, we convert and use as a feedstock to produce microbial ingredients such as yeast. This implies acid or enzyme assistant hydrolysis. And the process is similar to what I've shown you before. And what you have then left over, you can use then as organic fertilizer. Thereby you generate this circular economy and nothing is wasted. So a little bit about how we work then with these biomasses. And here you can see a picture of our biorefinery laboratory at our university. Where we are producing yeast on a small scale from the spruce tree sugars or the seaweed feedstock. And what is important first is to find the right combination of the sugars and other nutrients so that yeast can grow fast. The second thing that's important is to find the right type of yeast that grow well on this substrate with high yield and high nutritional value. And the third thing that's also important is the fermentation condition that needs to be optimized and the downstream processing that needs to be optimized to ensure high nutritional quality and also functional properties such as effect on health. So we have been working quite a bit with these protocols, and over time we have developed this. And the next step is then to upscale this. And that we do together with our industry partners. Overland is located just south of campus. They have access to much larger fermenters. So this gives us very valuable information about how these processes work on a larger scale. And it gives us yeast to test out in diets for fish and other farm animals. But I would also like to mention that last year we reached a milestone where we're able to upscale the production of yeast based on all the work we did in the biorefinery lab to a larger scale, where we produced 1,600 kilo of yeast from the sugar, 3 sugars. Here, Bolgo in Norway produced 7,000 liters of sugars that we shipped to Estonia, where Lallemann has their factories, and they produce yeast that was shipped back to Scandinavia, Denmark first, where they produced the fish feed. And then we ran a big field trial together with BioMar in their sea site up in northern Norway. This trial was just finished, and it gives us very important insight on how these novel ingredients work under field conditions, how they affect the growth performance, health. They will tell us a little bit about the sustainability and the economy of using such novel ingredients in these fish diets. We don't have results yet, but they will come soon. And then let's, yeah, okay. A little bit about yeast. So what is yeast? It is a eukaryotic single-cell organism that belongs to the fungi kingdom. And there are many different yeast species, over 500. And within each yeast species, there are many different strains. And but only few of these are approved to be used in feed in the EU. Use of feed in animal feed has a long history, but they've been mainly used as additive to improve health and performance, growth performance of the fish. The most common yeast species that is used in the industry is Saccharomyces cerevisiae. But in our center, Foods of Norway, we are working on producing the non-Saccharomyces yeast, and we use it as a protein source rather than a feed additive. And we only work with yeast that is already EU approved. And when you look at the composition of yeast, it is a protein source of high nutritional value. It contains about 50 and up to 58% protein and has a favorable amino acid composition compared to the fish requirement. It tastes good, especially when it comes from these sugars, 3 sugars. And it has also a positive effect on health. And it does have a low carbon footprint. And here you can see the amino acid composition of the yeast compared to high quality fish meal. And here you can see it's a favorable amino acid composition with the exception of methionine, which is lower. But of course, you take care of that by optimizing the diets in a way to meet the demand of the fish. We have, together with the University of California, looked at the LCA of yeast, or the climate footprint of yeast compared to soy protein concentrate. Also here we compared it to bacteria meal. And in the gray, you see the footprint of the soy protein concentrate, and in the green, you can see the footprint of yeast. And it does show that the yeast has a positive effect on the footprint, especially with respect to land and water use, marine and freshwater eutrophication, as well as greenhouse gas emission. And this was compared to yeast that was produced from sugars from wheat as a byproduct from the bioethanol production. We have run a series of experiments with yeast in diets for salmon in both the freshwater phase and the seawater phase, and with good results. Here I wanted to show you that we tested 3 different yeast strains or species with a high quality fish meal diet. And we looked at digestibility. And what you see here is that there's a large difference in digestibility. Two of the yeast strains, C. jardini, Chloromyces, had a high digestibility, but while Saccharomyces had a low digestibility. And this is due to the poorly digestible cell wall component that binds the protein in a matrix that are unavailable to the digestive enzymes. But we took this further into a trial with fish, fed the fish for 3 months, and here you see the result on weight and the feed conversion ratio. And you see that two of these candidates perform very well with respect to weight gain and feed conversion ratio, while again, the Saccharomyces didn't perform quite as well. So we decided to continue working with the C. jardini, or also called the Candida utilis, as a model yeast species in our center. We also been working quite a bit with documenting the value added of effects of yeast, especially the effect it has on health, because yeast per se cannot compete with the low prices of the soy protein concentrates. So we're looking for additional effects beyond the nutritional value. How does it affect health? Because that will increase the overall profitability. And yeast contain a wide range of different bioactive components that can have health effects. Yeast is composed of insoluble cell wall component and the soluble cytosolic component. The cell wall makes up about 30 to 50% of the yeast cell, and it contains, it has an outer layer with the manoprotein and the inner layer with the complex beta-glucans. The cytosol contains more the protein, the amino acid, the peptides, and some enzymes. The different yeast species varies a lot according to the cell wall composition and morphology. And again, this will reflect on how it affects the nutrition and the health effects of the animals. So what we're doing then, or here you see the different structures of these cell wall. So what we're doing is that we're evaluating these yeast products in first lab scale trials, where we use, it would be cell lines or cells, immune cells that we isolate from the salmon. And we look at the immune responses when they're exposed to these different yeast products in relation to selected biomarkers, both on the gene expression level and protein production level. Then the best yeast products we take further and evaluate in the fish experiment. And here we're using traditional methods together with a lot more advanced methods that gives us an in-depth understanding how the different yeast species affect the performance as well as the health effects of the yeast on a local level in the gut and also in the systemic level. I'll give you some examples of that. And in our study, we also used the soybean meal-induced enteritis model. The idea is that when you feed a high level of soybean meal to salmon, they will develop inflammation, referred to as the soybean meal-induced enteritis. The thought here is to simulate what's happening when you transfer the fish from the fish meal-based diet to the plant-based diet that can lead to this inflammation. And then we see how these microbial ingredients affect the enteritis. Are they able to prevent it? And in the picture here, you see a normal intestine, distal intestine, compared to the inflamed intestine. Quite a distinct difference. So in this experiment, we studied the effect on the different yeast species on gut health and immunity. We used soybean meal as a positive control, fish meal as a negative control. And we then had 3 test diets where we combined the yeast and the soybean meal. Same yeast as you've seen before, C. jardini, Chloromyces marxianus, and Saccharomyces cerevisiae. And what we saw on the histology level is that when you added C. jardini in the diets with soybean meal, it counteracted or neutralized inflammation. Chloromyces partially reduced inflammation, whereas Saccharomyces didn't really show much of an effect. We also saw that the soybean meal led to a large shift in the gut microbiota population with increased level of firmicutes, whereas when you fed the yeast, you saw more increase in proteobacteria and less of the firmicutes. And we also know that changes in the gut microbiota can affect the inflammation. And we also looked at the gene expression based on in the distal intestine, here based on microarray. And here you can see a heat map of different pathways related to different functions such as immunity, tissue repair, metabolism, detoxification. And blue here is downregulation of the gene sets, and red is upregulation of the different pathways. And I don't want to get into details here because you can read the article, but what you can see is that fish meal and the C. gendini led to a similar pattern. Chlomyces maxianus was in between, while the Saccharomyces cerevisiae and the soybean meal also led to a similar pattern, but opposite of the fish meal. So taken together, it shows that the C. gendini were able to neutralize the effect that we saw on the soybean meal on the inflammation. Chloromyces had some effect, and Saccharomyces in this trial didn't really have much of an effect. So it shows that yeast is has a positive effect on growth performance as well as health. And it is therefore an interesting alternative. But it's also important to know that the nutritional value of the yeast depends on the type of yeast you use and also what kind of downstream processing you apply. Here I wanted to share you another example of a trial we did. Here we used yeast in the diets of the salmon during the very critical period of seawater transfer. And this is where we are experiencing large losses in the industry in Norway. So we wanted to see if yeast could help alleviate this and make and develop a more robust fish, you could say. So we have two groups of fish, one fed the control and one fed the yeast during the freshwater stages. And then after 4 weeks of the freshwater stage, we took the fish and transferred them to the seawater, where half the control fish were fed, continue on the control, and where the rest was fed the yeast, and the same for the yeast group. Half was switched to the control and half continued on the yeast. And what we saw in this study is that the fish that were fed the yeast, they ate better, they grew better, and they were more robust during the seawater transfer with respect to histology and in the high gut. And also when you look at the protein production, we saw that you have reduced inflammation in the distal intestine. Here you can see the with respect to TNF alpha, interferon gamma, EL1 beta, and so forth. So yeah, we see that you reduced inflammation. On the gene expression level, it also looked like these fish were better adapted to the seawater transfer. So again, here you have a high quality protein source with functional properties that can help the industry alleviating the challenges associated with the seawater transfer. I mentioned we work with downstream processing of the yeast, which is important because it has an impact of the nutritional value as well as the functional property of the yeast. And we also basically here know that some of the yeast has low digestibility because of the cell wall components. So in this study, we used Saccharomyces cerevisiae as a model yeast. And we exposed the yeast cream to different downstream processing methods. This included cell crushing at different speeds, autolysis at different time spans, and spray drying at different temperatures. And when you treated the yeast and you look at the microscope, you can see clear differences between the intact whole yeast, those who were exposed to the cell crushing, and those who were exposed to the autolysis process. And you see large differences on the cell structure compared to the cell when exposed to cell crushing. But what was interesting is that when you look at the digestibility here determined in salmon, you saw the best effect on digestibility was when the yeast was autolyzed. This is where the native internal enzymes within the yeast cell starts breaking down the cell membrane structures and it liberates the protein. So you increase the digestibility. And because of this study, we decided to continue with downstream processing by autolysis because it was the most efficient method, but it was also the cheapest method. And here we used the 3 non-Saccharomyces yeast, C. gendini, B. adnivorans, and Vicomyces anomalus. And we exposed them to autolysis for 8 or 16 hours, and we dried them then at 180 degrees Celsius. And when we look at the microscope, we saw big differences in the internal cell membrane structures. And if you look at the confocal microscope images, you see the location on the mannoprotein located on the cell wall surface of the cells, yeast cells, here shown in green. So what happened is when they were exposed to the autolysis, we saw large differences in the cell wall thickness. You can see there's large differences among the different yeast strains tested, but we also saw that the autolysis reduced the cell wall thickness. We also saw large differences in the mannan and glucan content among the yeast, and we saw low content on mannans in the C. jandini, but the glucans were high, and the opposite was the case for Vicomyces anomalus. Autolysis reduced the glucan content, but didn't have such a large impact on the mannan in this study. The digestibility study with the salmon, and we looked at what happened when you exposed the yeast to autolysis. And you see that the protein digestibility increased in all yeast except for the last one. But the effect wasn't as great as we've seen before, because here we froze the yeast cream after it was fermented. And I think that led to inactivation of some of these enzymes. But at least you can see there is definitely an effect here. Then we go on, and we had then with the use of atomic force microscopy that has a functional tip, combined with specific antibodies that bind to these beta-glucans mannans on the surface of the cell wall structure, we can map out the properties of the cell walls in terms of binding properties and the structures of these components. And this tells us a little bit about the nutritional value and how the processing by autolysis affects these values. And it also shows that these properties are correlated with immune responses that we see in the fish. So basically, we ran a trial where we fed the fish control-based diet based on high-quality fish meal, positive control based on a high level of soybean meal to induce inflammation, and then 3 yeast diets where we combined the yeast with the soy. And we fed the fish for about 37 days. This was all done in fresh water. So what we see here is that especially the C. jandini yeast and the Vicomyces anomalus led to a reduction in the mild inflammation that we saw with the soybean meal. But we also saw the effect on the histology was much greater when we actually had exposed the yeast to the autolysis. But when we correlate the different diets based on selected immunobiomarkers, we saw that there was a strong positive correlation between the fish meal and the autolyzed C. jandini yeast. When you look at correlation between the soybean and the other diets, you saw a negative correlation. It suggests that especially autolyzed C. jandini was able to reduce the inflammation and led to a similar immunoresponse as the more neutral fish meal diet. In the same study, we evaluated the immunomodulating effect of the diets on a systemic level, where we used the spleen as an immune active organ. And here we only compared the fish meal, soybean meal, and the 2 diets with the C. jandini yeast. And we basically characterized the immune responses based on both the gene expression response and the protein production response or level. And we do see that soybean meal led to systemic inflammation. And we also here see that the C. jandini prevented inflammation. But this, I don't expect you to follow all these details. You can go into the article and read that later. But this, you can see a network of different gene function and how the different diets are related to these. And if you compare the fish meal group to the soybean meal group, you see that soybean meal led to a large differences in gene expression and function related to inflammation. And when you looked at the fish meal compared to the autolyzed C. jandini, you see a similar gene regulation, meaning that the autolyzed C. jandini was able to control the inflammation caused by soybean meal by activated biological pathways related to endocytosis and a range of different innate immune responses. But when you look at the protein level, we saw that the autolyzed C. jandini controlled the inflammation that was caused by soybean meal by decreasing pro-inflammatory cytokinins and basically increasing the anti-inflammatory responses, which led to immunohomeostasis. So while the intact CGD didn't really have this same effect on the gene expression. So here we have correlated again the different diets based on selected immunobiomarkers. And we see that the fish meal and the outlying CGD has a positive correlation. And it does suggest that when you feed 5% outlying CGD, you prevent the inflammation caused by the soybean meal, and you are able to maintain the immunohomeostasis. So again, showing that this yeast is bioactive, and it is also potential as a sustainable protein source in aquafeeds. So basically, now I have shown you how we work along the value chain, all the way from the biomass throughout the value chain to the final fish product. Together with the industry partners, we are also looking at LCA evaluation of the yeast from the first generation sugar, tree sugars, and we've seen interesting results. We also look at techno-economical aspects. How efficient is it? How cost efficient is it to use these ingredients in the diets? And the next step is scaling up. But when we see the results from the techno-economical analysis, we see that the sugar is the biggest impact factor that affects the overall cost. And since we started the center in 2015, the sugar prices has actually doubled because of the increased demand for biofuel. So there is clearly a feed fuel competition here that drives up the sugar prices that also affects our possibility for upscaling. But because of this, we are working on an alternative strategy, and where we are working with different type of microbes and different forest streams that are cheaper in order to reduce the cost. So in Foods of Norway, along with this spin-off project, Nordic Feed, we are doing this, where we are looking at other waste streams from the forest. We're working with filamentous fungi. They can produce high level of protein, 60 to 65%, based on cheaper sugar streams. We're also looking at other type of waste streams from food waste and also from slaughter byproducts, fat-rich slaughter byproducts, hoping that this will lead to a profitable development of microbial ingredients that can be upscaled. We applied for a new project called Forest Feed, where we're going to take the results from the Nordic Feed and Foods of Norway and upscale it, so we can hopefully be able to scale up the production of this filamentous fungi. So let me see now. I have been talking for a while here, and I don't think I'm going to go into details about the insects, except for mentioning that we are working with insects. We see them as interesting. We have 4 PhD students working with insects, and it's definitely also a very interesting alternative. But I think with the time being here, I will go to the last slide, which is about the future. So when we look at the future, I think we will see more competition for our natural resources, driven by factors such as population growth, development of the bioeconomy, and climate change. And this will lead to a greater competition between the use of biomass for food and non-food purposes, such as for bioenergy. And this will definitely increase the demand for the biomass. So thus, I expect to see that we will have increased production of the novel ingredients, such as the microbes, but this will be based on different input factors, such as increased use of different waste streams as well as waste gases. So basically, but the thing is, before we can replace large commodities such as soy and fish meal, we need to upscale the production of these microbes, and that will take time. But in the meanwhile, we have to definitely look at what we have available to us today and maximize the use of existing ingredients. And we also have to take a new look at the legislation so we have access to more of the substrates. So with this, I think I will thank you for your attention, and I will try to stop sharing with you. Yeah.
Thanks, Margaret. Very great presentation, very insightful research you're doing in Norway. Kabir is here. He managed to join. That's from Philippines. Before we start the discussion, should I go through the questions or would you like to discuss first, Kabir and Albert?
No, I think
No, I
I'm
think
just
questions will be.
Okay,
yes.
so
No.
there are two questions. One is from Pamela Pardee. So she says, I'm impressed with the technology available to convert trees to high quality protein. But how much of this ingredient can be produced without running the risk of causing environmental damage, such as deforestation? Where do we draw the line for sustainability?
Yeah, because this is a very good question. We are only basing the process on forced byproducts that otherwise can go to waste. The other thing is that the harvest in Norway is all based on sustainable harvest, and we're taking out a lot less than the increase in the biomass. So therefore, it is done in a sustainable way. And when you cut the forest, you also replant. This is the law in Norway. You want to have the trees growing, and thereby they do bind the carbon and they store it, you know. So it's important when it comes to CO2 binding. And when it comes to the amount of biomass available, we have enough biomass available to serve the aquaculture industry in Norway with 10% yeast inclusion. But of course, right now, the problem is there is large competition for the biomass, for biofuel, for other purposes. And this is a political question. How do we use the biomass the best way? How can we then use it for protein, for food instead of biofuel, for instance?
Yeah, I think there are many players involved, so it goes to
Albert talked
politics and
food
yeah.
versus fuel, I guess. Food
Yeah,
versus
Albert has a
No,
question.
no, it was just to say that I have done some work for a company in the US that produce phytosterols, basically from spruce. They are the largest producers of phytosterol in the world. They are used at the moment in the margarines in the that we consume in Europe, and they're used as a way of reducing the cholesterol level within humans. I've also used them as a possible replacement for dietary cholesterol for shrimp. As we know, cholesterol is an essential nutrient for shrimp, and we have shown that phytosterols from spruce, again, the company is called Arboris, can actually reduce the requirement for cholesterol by shrimp. And also, one of the things about shrimp is that it does have a higher cholesterol content compared to other products. As we all know, it's the number one consumed commodity in the US in terms of seafood. But cholesterol is very much on the radar screen of many consumers. And so again, I'm just putting out agreeing with Margareth, and that is that spruce can produce many different types of products, and one product that is currently being used now are the phytosterols.
I have some specific general questions, and then I will go specific if you permit, Margaret, right? So first is why choosing wood biomass, right? We know we discussed about it. And then why yeast? That's one thing. And then why the species that you have chosen? That's the first question.
Yeah, good question. The biomass is chosen because we need to see what we have available in Norway, because I mentioned 3% agricultural land, only half of that can be used to produce crops. So
Yeah.
the land area and the plant protein sources are very limited. This is why we import so many other ingredients. But we are in a good, fortunate situation where we have access to all these bioresources, with all the trees, with all the seaweed, with all these other marine species in the sea. I didn't mention tunicates as such, but we also work with those, you know. So this is why we are looking into biomass. And we have technology today that we didn't have some years ago. The enzyme technology that allows us to break down these biomasses and build them up again and convert them into microbes, you see. Why yeast? Well, we also work with bacteria. And we work with microalgae as well as the yeast. Our first experiments showed very good results with the yeast, 58% protein, high digestibility, and very good health effects. So we thought yeast is the way to go. But over the years, we see that yeah, the sugar is expensive, so we need to go into the filamentous fungi. They have higher protein content even than the yeast, and they are cheaper to produce. So this is where our focus are now. But the technology is similar. So we have a platform that we develop further. I do believe that the bacteria is important because they can use the waste streams, the hydrogen, the CO2, the methane. And we actually now got a new project where we're also going to look into this. I'm very excited. We've been working with the bacterial meal from the natural gas, but now we can also use the waste gases. This basically means that we're less dependent on photosynthesis and less dependent on agriculture to produce our future feed. Of course, there are issues there too. There's a lot of water, there's a lot of energy needed to dry it, but we need to get there. We need to use those resources.
Okay, can I go to more specific, Fanny?
I think so. We have other two questions, but three questions.
Okay, what is like about the gene expression, right? And
Mm.
you have shown that the species that you were working with, the response was similar to fish meal, whereas Saccharomyces cerevisiae is kind of similar to soybean meal, right? In terms of inflammatory responses. Why do you think Saccharomyces cerevisiae was similar to soybean meal, number one? Is it because how they were processed, or just I don't know. I don't know. Just you can speculate,
Yeah.
or you can.
Yeah,
Sorry.
it is basically, I think a lot of it has to do with the way it was fermented and how it was processed, because we know that Saccharomyces has been used as a feed additive for many years. So yeah, so I wouldn't conclude based on one experiment, but it was interesting to see how distinctly these different yeasts reacted in terms of immune responses.
So final one. Sorry, Fanny.
No
The
problem.
final one. The final question is, if we use like autolyzed yeast, or as a or I say, but you say crushed, I say broken cell wall, or whole yeast. Instead of that, if we use a purified component, right? Because we know beta-glucan, MOS, chitin, right? Nucleic acid. If we use purified component, what would be the? Do you think that there will be some different response than the whole yeast or autolyzed yeast?
Yeah, I do believe so, because right now we are focusing on finding alternative protein sources. So we're using the intact yeast or the autolyzed yeast. So that's important. However, there are a wide range of different yeast products out there where you're separating the cytoplasm from the cell wall, and they can have very unique immune responses depending on what yeast you're using. Saccharomyces given some responses, other yeast might give other responses because of the composition and the properties of the cell wall. We are working with yeast as a feed additive too, by the way, during the seawater transfer, and we do see a very interesting responses. But yeah, our purpose is to find these alternative protein sources.
So we have two, well, it's more general questions. One is from Sofia Moraes. She asks, she says, amazing presentation. Thank you. And in your opinion, what will be the main constraint for upscaling and mass production of microbial biomass for animal feed?
The main constraint is that, okay, I said that we've done LCA on the yeast from the spruce trees. It actually is even better than the yeast from the first generation sugar, the wheat sugar. So we have very good numbers when it comes to LCA. Some of the yeast that we work with has a protein content of 50 to 55. That is, I would say, too low for a salmon. We're working now with newer ingredients with a protein content that can be up to 65%. That is more important. But scaling up, you have to show that you have a high level of protein. It has to be digestible. You have to show that you have value-added effects, such as the health. In addition, you need to show that is a positive effect on the economy, the techno-economical analysis. When you have this ready, which we're working on, then the investors will be there and they will want to build factories. And we have investors showing a lot of interest, but we need to show good numbers.
Okay. Albert, do you have any? Yes?
Yeah, I mean, just to add to that, in the late 70s, early 80s, a company called EWAS produce a diet called C10, which was basically 95% Candida lipolytica as a feed, as a larval feed for carps, for more omnivorous species. But also, I mean, to ask about scaling up, also in just showing my age, in the late 70s, early 80s, I did some work for ICI in the UK that were producing Methanobacter. It was originally as a protein source for human consumption. But what they didn't realize in those days is that humans, we cannot process nucleic acids, we have uric acid accumulation and gout. And so I was given the task by them to feed different levels of the nucleic acid portion of the bacteria to trout to see if they could metabolize it. And of course they could. But again, the beautiful thing about bacteria or yeast is their doubling time, that you know, you just need the substrate and you can really produce the product, you know, where you need it. And again, one of the nice things that Margaret said is that, you know, she mentioned that Norway is such a great success story for many countries, but it's really like 95% of its ingredients are imported. The problem is not today, it's tomorrow, and what's going to happen with feed ingredient prices. And so, and this is also very common in Asia. For most Asian countries, they have to import. So it's all about how we can better use the resources that we have in our country. Anyway, sorry, that was just a comment, but
Yeah, because we have looked at how the salmon can cope with the nucleic acid in the bacteria and the yeast. And we see, for instance, with bacterial meal, we see they cope with it quite fine. They have the rate-limiting enzyme, uricase oxidase, that convert and metabolizes the purines, so, or nucleic acid. So no problem there. No problem in pigs, but in broiler chicken, there might be an issue. So in fact, nucleic acid is actually positive with respect to immunity, gut integrity. So there's this additional benefit, you know. But yeah, there are microbes that are used directly for human food too, and they're looking more into that. So that's another path, you know.
I think we have a very interesting journey ahead, right? I think the next 10, 15 years we will see
a bumpy ride, yes.
Yeah, things will change. Can we just take 2 minutes, go away of what we are talking about? I was looking at different companies establishing in Singapore or other countries, right? Alternative proteins, right? Meat proteins, new meat, etc., right? Just a question, right? Where are we going with this, right? Is the industry is going to change completely in the next 5, 10 years? There's a prediction that 90% of dairy farmers will lose their, farming in the next by 2030 to the alternative milk. So are we as aquaculture community feeling any threat or opportunity? It's a very general question, but we need to think today, right? We are talking about sustainability issues, but things are changing very rapidly in the coming years.
To comment on that is a good question too. I think we're going to see big changes coming. Like I mentioned initially, with the climate, with the war situation, pandemic, it changes the world. It's going to change the way we're going to feed our animals. But already today they're seeing an increase in the aquafeeds by 20 to 30% in Norway, by the way. It's probably going to get worse next year. So there's a very different situation. And this is why we need to think about what do we have in terms of existing ingredients, the byproducts, the plant byproducts, the animal byproducts that don't compete with human food. We need to use them in a better way than we are doing today. They are there, they're abundant, it's a kind of an easy step. Parallel with that, we need to also, of course, develop these microbes. So, I believe they're going to come. I believe they're going to be important. In addition, I really think that in Europe and Norway, we need to take a new look at EU legislation. It's based on old methods, legislation from 30 years ago. The substrate that are available, we cannot use today to produce insects or microbes because of these regulatory constraints. But with these new methods, we can prove and document safety in a whole different way, And the other thing I want to mention also, yeah, it will probably change the way we're thinking in terms of GMO. GMO and plants that are able to cope with the climate changes. So yeah, I see a range of different things going on.
Albert?
Well, I agree. I mean, I like to probably make a plug for seaweed products. I mean, certainly I've been working with companies for over 10 years that basically process macrocystis and turn it into a hydrolysate, which we then use in, especially in shrimp feeds, but as a, not only as an immunostimulant and binder, but also for the source of minerals that they contain. Again, seaweeds are sea vegetables, like you say, you know, I mean, again, there are many species. The fatty acids that are present in them tend to be of the omega-3 series more than the other series. Again, this, I mean, we have, I mean, but I think, you know, one important point, I mean, Margaret touched on it a little bit, but the largest source of animal protein available to feed compounders are the byproducts from the production of poultry and pigs and ruminants. Of course, we have the issue of prions and mad cow disease and the issues particularly with ruminants. But when it comes to poultry byproducts, you know, it is the largest source of material available to the animal feed compounder, and it's certainly in Asia it's used, and in Latin America it's used. In Europe we have other issues, like you say, you know, we have issues to address at the legal level. But certainly the ingredients are there. But the future is not using things that can compete with humans. We have to use feed grade to produce food. We cannot use ingredients which compete with human consumption, I think, in the long term. Anyway, that's on my own personal view.
Can I ask another question that we did not get to? I think Margaret didn't get to present. Insect meal, right?
Yes.
So much money is like flowing into insect meal. Almost more than $400 billion. Over 400 companies now working on insect meal. Where are we going? And how long will it take? Basically, you know, like it's now everywhere, cost prohibitive, not standardized. How long will it take for this product to come to play for mainstream aqua feed?
Well, I know there's a lot of interest in insect meal, especially black soldier flies. I was just at the ISFN meeting in Toronto, and a lot of the presentations there were based on results from black soldier fly. How long will it take to scale up? Well, today, again, the production volume are too low to compete with fish meal and soy protein concentrate. Insect meal is still too expensive, 2 to 3 times more expensive. So the industry is using it, but maybe at the low level, maybe 2% or so. And again, you know, to upscale the production, it's about access to these substrates. Because if we can use these alternative substrates and document the safety, we can realistically upscale the production that they can make a difference. But insects alone will never be able to solve the whole issue. We need to work in parallel with the microbes, with marine species on a lower trophic level, and we need to use the byproducts from the agriculture and aquaculture and plants, you know, like was mentioned earlier. So how long time will it go? I don't know, but there's a lot of interest also in Norway, you know.
Sir Albert.
No, again, yeah, it's, we always say to produce feed for a day, it's easy. Doing it 365 days a year of consistent quality, you know. Aquaculture feed is about 60 million tons of feed. Norway, you said 1.6 million tons. You know, and we have to, at the moment, the price of the insect meal, it looks nice, it looks interesting, but like you say, until we have volume and we can start reducing the price, it's not really going to be used by people. But one of the interesting things is that the choice of substrate really can affect the nutritional composition of the particular insect that you're growing. And if you use a marine substrate, you can really improve the omega-3 content of the animal. And I think that's interesting because, again, Norway has lots of these, waste streams that we can really use and to improve, produce a product which has got a higher value anyway.
Yeah, yes, definitely. They are interesting. The fish byproducts and the seaweed, for instance, would increase the omega-3s. So that would be basically taking care of that problem, Mm.
Thank you. Thank you. I think Fanny has lost her video connection. She's coming back. Okay,
Yeah,
thank you,
sorry,
Fanny.
I'm having very bad connection, so I'm on and off.
The topic is
Yeah, so I still have 2 questions from the audience. Then it's a bit more. So we are going back to the bacteria, the yeast. So one is from Simab Zira asking, would like to know the pro-inflammatory and the anti-inflammatory gene response versus the yeast protein.
Yes, the pro-inflammatory was TNF alpha, interferon gamma, and it was, but this is on the protein level. We have specific antibodies from salmon, but and then we have the IL-10, which was the anti-inflammatory response that we used here and as an example when we looked at the correlation.
Okay, thank you. Another one from Yu Genhua. This one I think is a bit difficult to reply, but I think we all can try. So how long will it take to make bacteria-based proteins profitable in the aquaculture industry?
Well, the thing is, we've been working with bacterial meal now for several decades, including us, and it is very nice to see that our innovation is now taken further into commercialization. And I just talked to Calista in Sorrento last week, and they said that in 4 months, they have the first factory ready to go in China. They're planning on making other factories, maybe in the Middle East and South Americas as well, and maybe also this part of the world. But so we're talking very close right now. But I know there's a lot of activity around the globe, producing also bacteria from waste gas. Not such a high volume yet, but they have upscaled it. I know that's going on in China, for instance. So yeah, it's coming,
I think Albert has some comments.
No, I agree. I mean, it's coming. It's coming.
If I may update on ANN a little bit, right? Get 2 minutes from yours, Fanny, with permission.
Yeah, sure, no problem.
I think 6,900 members, right? As of today, almost 6,900 members, and I think we are. I would like to thank Margaret personally, right, for very wonderful and insightful presentation. I think there's a lot of interest into what you have presented today. Yes, that's from my side personally. Thank you.
We have another, maybe the last question. It's from Pamela Pardy again. She says it's indisputable that insect meal is nutritious. Why use fish byproduct as a substrate when it can be fed directly to other animals? So she says just another conversation.
Well, definitely we need to take the fish byproducts and use them for human food. Number one, we need to use them and incorporate them back into the fish meal. Number two, but there are byproducts that might not be so suitable for these purposes and that are today wasted, and then we can use, for instance, to feed the insects or as enriched nitrogen sources for the microbes. It's all about using the byproducts in the best way. But today, there are still a lot of marine byproducts are wasted, go to the sea because of the boat, might not be able to take care of all these byproducts. There's a lot to be done in that area, which would be a quick fix to the raw material crisis, you might say.
Okay, so I think we are coming to the end of this session, of today's session. Last comments from everyone?
I could also have a last comment that what I presented today was based on a lot of work with a big team of Boots on Norway. I gave you some examples of the references, but I also provided a reference list for those who wants to dig further into it. So yeah.
Yeah, I mean,
So
would just like to
sorry.
say, to add that, sorry, that Norway as a country also really, really supports, that salmon is a very valuable export for the country, and what they do is part of the export revenues are actually recycled back to support the industry in terms of research, in terms of universities, and again, it's very, very important, and it really is a great example to other countries.
I think Fanny is disconnected again. So if Fanny doesn't come back, just if Margaret can send like Rongshin, I'd also like to thank Rongshin, right? Rongshin has been working in the back to make this a successful program since we switched to with Aquatic Innovation Center in Singapore as our support organization. I would like to thank Rongshin as well. And thank you, Margaret, again. And if you can provide the list of the references, right? If not the PDFs to Rongshin, we can share with our audience, and also our members.
Yeah.
And Annie, you can be the closer. Thank you.
Sorry, today very bad connection here. So I would like to thank again Dr. Margaret Overland for joining us today, giving this great presentation, insightful ideas and innovations you're doing in Norway that the rest of the world can also learn from you. And also thank our usual panelists, Kabir and Albert, for joining us different time zones. And rushing from work and also a bit fluid, but joining us. So everybody doing the extra mile to be here. So so please, we will send you the audience a feedback form. So for those who can fill up and return to us, we will send you the presentation once it's available, and also the video will be uploaded in the ANN YouTube channel in a few days or next week. So we are preparing another session for the coming future, maybe next month or the other. So stay tuned. We will contact you or we will send the EDM. So please join us again. So until then, have a good day. See
See you
you
next.
again. Thanks, everyone.
Thank you so much.
Thank you.
Bye bye.
Thank